Photosynthesis starts when the sun powers photosystem 2 to release 4 electrons. These electrons continue to replace the electrons lost by photosystem 1. The electrons from photosystem 2 are also used to bring hydrogen ions through the thylakoid membrane, so there's a higher density in the thylakoid space. The high density of hydrogen ions in the thylakoid space causes them to go through the ATP synthase and produce ATP. The electrons from photosystem 1 combine with NADP+ to form NADPH. ATP and NADPH continue on to the light-independent reactions. Because photosystem 1 got its replacement electrons from photosystem 2 needs to replace its electrons. It does this by splitting apart water molecules and using their electrons. The Calvin cycle starts with RuBp and CO2. The CO2 connects to the RuBps to make a 6-carbon molecule. Then the 3 6-carbon molecules split to form 6 3-carbon molecules called PGAs. The low-energy PGAs are turned into high-energy PGAls with the power of ATP and NADPH. This reaction results in the production of an ADP and an NADP+.
Tactical nuke incoming!
One of the 6 PGALs is taken from the cycle each time to start forming a glucose. The other PGALs are powered by ATPs, so they can reform into RuBps to restart the cycle. The cycle must be repeated twice to form one glucose because each time it only gives off one PGAL. Glycolysis starts with a single glucose. 2 ATPs then put their power into the glucose and give off 2 ADPs. This is called the energy investment. Then 4 ADPs take power from the glucose and give off 4 ATPs. Also, 2 NAD pluses combine with hydrogen ions to take energy from the glucose. This reaction produces 2 NADHs. The ADPs turning into ATPs and the NAD pluses and the hydrogen ions turning into NADHs is called energy recovery. After the energy recovery, 2 pyruvic acids are produced. The 2 NADHs produced can be used in fermentation or the electron transport chain. The 2 pyruvic acids that were produced can either be used in the Krebs cycle or fermentation. The Krebs cycle starts out with a single pyruvic acid. This acid can change into an acetic acid by giving off a single carbon dioxide. This reaction changes a NAD plus to an NADH. The acetic acid then joins with a 4-carbon chain to form a citric acid. Citric acid then gives off a single carbon dioxide to make a 5-carbon molecule, and then another carbon dioxide comes off to make a 4-carbon molecule. Then the molecule is rearranged multiple times by NAD pluses and ADPs and FADs to form the original 4-carbon chain that started the cycle. All of these reactions give off 2 NADHs, an ATP, and an FADH. NADHs from glycolysis are used in the electron transport chain to pull hydrogen ions through the thylakoid membrane. The NADH can pull 3 hydrogen ions through the thylakoid membrane. And FADH2 can also be used to pull hydrogen ions through the thylakoid membrane, but it can only pull about 2 hydrogen ions through. The NADH reaction gives off NAD+, and the FADH2 reaction gives off FAD. The high density of hydrogen ions in the thylakoid membrane causes them to go through the ATP synthase. When this happens, ATP and oxygen are produced. The oxygen combines with some hydrogen ions to form water. With glycolysis in the Krebs cycle combined, 38 ATPs are produced, but only 36 are gained.